Pathology. Tata Memorial Center. Understanding reticulocyte. Reticulocytes are young or juvenile, immature red cells, larger than RBCs, non-nucleated. They contain remnants of ribonucleic acid, RNA, protoporphyrin, ribosomes. Released from the bone marrow into the bloodstream. Late 1800. Paul Ehrlich Properties of Reticulum, senescent RBCs. 1865. Heinrich Erb, Granulated Erythrocytes, Transition of WBC to RBC. Early 1900. Theobald Smith Young Red Cells. 1922. Edward Bell, Term, Reticulocyte. 1932. Heimeyer-Westheiser Classification of Reticulocytes Maturity. Discovery of reticulocytes. Where does reticulocytes come? In bone marrow. In peripheral blood. Questions. Why reticulocyte count? Reflection of erythropoietic activity. To diagnose anemia due to ineffective erythropoiesis. To assess response to specific therapy, X, iron deficiency and megaloblastic anemias. Evaluation of bone marrow recovery, after bone marrow transplantation or intensive chemotherapy. To assess response to erythropoietin therapy in anemia of chronic renal failure. Specimen collection and storage. K2-EDTA is the anticoagulant of choice. Venous or capillary blood. At room temperature within 6 hours after blood collection. If sample analysis is delayed, the sample should be refrigerated. Samples stored at 2 to 6 degrees Celsius may be stable for up to 72 hours. A group of test tubes with labels. Description automatically generated. NCCLS, CSLI Guideline 44A2 Vol. 24 Number 8, 2004. Reticulocyte count methods. 1. Manual. Supravital dye refers to staining of cells in a living state before fixation or drying or with passage of time. New methylene blue. Brilliant cresyl blue. Azure B. Fluorescent dye. Acridine orange. 2. Automated. Cell counters. Flow cytometer. Quality control. Weighing balance and pipette should be calibrated. pH meter should be calibrated. pH of buffer should be checked before use. For cell counters, 3 level of control should be run. For better and more reliable results, use a new methylene blue because it stains more deeply and more uniformly than brilliant cresyl blue. Azure B is a satisfactory substitute for new methylene blue. Reticulocyte count by supravital dye. Staining solution. 1% brilliant cresyl blue or 1% new methylene blue. Dissolve 1 gm of BCB, NMB in 100 ml of isoosmotic phosphate buffer. Isoosmotic phosphate buffer, pH 6.5. A. Na2Po42H2O, 150 millimoles, L, 23.4 gm, L+. B. Na2. HPO. 4. 150 millimoles, L. 21.3 gm, L. Mix 64 milliliters of A plus 36 milliliters of B. Check pH. Filter with Wattmann's number 1 filter. Keep in dry bottle. Stable for 4 to 6 weeks. A hand holding a beaker with blue liquid. Description automatically generated. Principle. Microscope. A. Equal volume of blood collected in ETA are incubated at 370 C. With supravital dye staining solution which stains granules of RNA in red cells. A thin smear is prepared on a glass slide from the mixture and reticulocytes are counted under the microscope. Number of reticulocytes is expressed as a percentage of red cells. Procedure. Mix equal quantities of blood and dye. Solution. A larger proportion of anemic blood, and a smaller proportion of polycythemic blood, should be added than of normal blood. Incubate at 370 C in water bath for 15–20 minutes. Resuspend the red cells by gentle mixing. Make films on glass slides. When dry, examine the films. Under the microscope using oil immersion objective. Reticulocyte after supravital staining. The reticulofilamentous material stained deep blue. Any non-nucleated red cell containing 2 or more granules of blue stained material is considered as a reticulocyte. The College of American Pathology, ICSH. Reticulocytes appear as polychromatophilic, gray, blue, cells. Reticulocytes in Romanovsky stains. Calculation. 2 commonly used methods for counting reticulocytes. Standard counting procedure. Miller reticle procedure. Standard counting procedure. Dry smear is examined under oil immersion. Total of 1000 RBCs are counted. Number of reticulocytes are recorded per 1000 RBCs. Calculate the percentage of reticulocyte and absolute reticulocyte count. Reticulocyte percent equals number of reticulocytes counted. 1000 RBC, number of red cells counted, x 100. Miller reticle procedure. Miller ocular disc is used for counting of reticulocytes. The Miller ocular disc contains 2 squares, A and B. Square A is 9 times larger than square B. Square B is used for counting red cells, while reticulocytes are counted in square A. Reticulocyte count percent equals total reticulocytes counted in square A times 100. Total red cells counted in square B times 9. Absolute reticulocyte count, retic percent times RBC, is the actual number of reticulocytes in 1 L of whole blood. Reticulocyte index, corrected reticulocyte count. When there is severe anemia, the reticulocyte count can be corrected and expressed as a reticulocyte index. Reticulocyte index equals observed reticulocyte percent times measured HB or PCV. Appropriate normal HB or PCV. Corrected reticulocyte count greater than 2% indicates reticulocyte release appropriate for the degree of anemia. If less than 2%, reticulocyte release is inappropriate. Reticulocyte production index, RPI, equals corrected retic count, percent, divided by hash days, maturation time. Stages of maturation of reticulocytes. Stage 1. Dense cohesive reticulum in non-nucleated red cell, 0.1% of reticulocyte count in normal individuals. Stage 2. Extensive network of loose reticulum, 0.7% of reticulocyte count in normal individuals. Heimer and Westheiser, 1932. Stage 3. Small reticulum along with scattered granules, 32% of reticulocyte count in normal individuals. Stage 4. Scattered granules, 61% of reticulocyte count in normal individuals. A close-up of a microscope. Description automatically generated. A close-up of a red circle. Description automatically generated. Differentiating between reticulocytes and other cell inclusions. Heinz bodies. These are precipitated globin chains attached to the red cell membrane. They stain light blue and are seen in glucose-6-phosphate dehydrogenase deficiency following exposure to oxidant stress. Pappenheimer bodies. These are iron-containing granules which appear as one or more small dots in red cells. A group of blue dots. Description automatically generated. A group of purple circles. Description automatically generated. HBH inclusions. These are round, golf ball-like inclusion bodies seen in alpha thalassemias. Stain greenish-blue. These can be easily differentiated from reticulofilamentous material. Howell-Jolly bodies. These are nuclear remnants in red cells seen in certain anemias and following splenectomy. A supravital dye stain both denandrina. These structures are also stained. Granules of dye, superimposed on red cells, if stain is not filtered before. A white surface with yellow dots. Description automatically generated. Reference range. Reticulocyte percent. Adults and children, 0.5–2.5%. At birth or cord blood, 2 to 5%. IV. Causes of increased reticulocyte. high altitudes, hemolytic anemias, blood loss. Following specific therapy of nutritional anemia, like iron in iron deficiency anemia, folate in folate deficiency anemia, with B12 in B12 deficiency anemia. Hemoglobinopathies, e.g. sickle cell anemia. Causes of decreased reticulocyte. Aplastic anemia and pure red cell aplasia. Bone marrow infiltration, leukemia, lymphoma. Myelofibrosis, metastatic malignancy. Renal disease. Anemia of chronic disease. Alcoholism. Myxedema. Ineffective erythropoiesis, megaloblastic anemia, sideroblastic anemia, thalassemia, myelodysplasia. Following blood transfusion. IV. Fluorescence method. Reticulocytes can be counted manually by fluorescence microscopy on appropriately stained films. Add 1 volume of acridine orange solution, 50 mg, 100 ml of 9 grams, L-nickel, to 1 volume of blood. Mix gently for 2 minutes. Make films on glass slides, dry rapidly and examine with a fluorescence microscope. RNA gives unarranged read fluorescence, nuclear material, deoxyribonucleic acid, DNA, fluoresce yellow. Although the amount of fluorescence is proportional to the amount of RNA. Clinical utility of reticulocytes parameters. 1. CHR. Reticulocyte cellular hemoglobin content expressed in Pg cell. Mean of the retic-CH histogram for the reticulocyte, which reflect the functional availability of iron for the cell and the cell incorporation of iron into hemoglobin over the last several days. Detects functional iron deficiency with more sensitivity than biochemical parameters. Recent studies evaluation iron status in young infant sensitive and specific marker for predicting later anemia. Helpful for early measurement. Frequent blood donors early detection of iron depletion. Signal. 2. Immature reticulocyte fraction, IRF. Previously used term reticulocyte maturation index, RMI. Population of reticulocytes most recently released from the bone marrow contains the highest concentration of RNA. IRF helpful in evaluating marrow erythropoietic response to anemia in patient receiving human recombinant erythropoietin, RUPO, or iron therapy. IRF earliest marker of marrow response. It increases before retic count, hemoglobin and hematocrit, RBC count. After marrow transplantation, an increased IRF has been observed as one of the first signs of cell recovery. NCCLS document H44 on CSLI. 3. Reticulocyte mean volume, MCVR. Decrease, iron deficiency, thalassemia. Depleted iron stores. Up pointing arrow when iron supplements are given. Down pointing arrow during iron deficient erythropoiesis. Increased macrocytic anemia. Rapid normalization with treatment inversion of MCVR, McRitio. Sickle cell anemia hydroxia therapy fetal HB synthesis increased hydration of sickle reticulocytes MCVR. Person with cane. Reference. Dassey and Lewis Practical Hematology 12th edition 2017. S.B. Mackenzie and L. Williams Clinical Laboratory Hematology 3rd edition. Clinical utility of reticulocyte parameters Elisipeva, MDA, Kalubrignara, MDB, Federicus Paleo, MDA, Mario Plebin MD. THANKYOU. Handshake.